So hi everyone, I hope everybody is doing well. I'm Justine, I'm a first year master's student working at the Douglas, and I'll be moderating the talk this morning. So it's my great pleasure to welcome Yashar Zeighami for today's CIC lecture. Yashar received his bachelor's degree in electrical engineering and his master's degree in biomedical engineering from the University of Tehran. He finished his PhD in neuroscience at McGill University in 2018, working at the Human Dopamine Neuroimaging Lab, studying Parkinson's disease progression via brain networks. Dr. Zeighami visited the Allen Institute for Brain Sciences as a visiting scholar during his PhD, and he did his postdoctoral fellowship at the McGill Center for Integrative Neuroscience. So thank you very much, Yashar, for being here today. Just a quick reminder to everyone to please keep your mics closed during the presentation, and I'll be monitoring the chat, so you can write your questions in there or simply raise your hand. And if it's okay with you, Yashar, we can take the questions during the presentation. I'll just find an opportune moment to ask them. And Dr. Yeah, okay, great. Dr. Zeighami has also agreed to hold a student discussion at the end of the talk for any more open discussion questions. So anyone who wants to can stick around for that as well. So with that, Yashar, thanks again for taking the time to give this lecture today, and the floor is yours.
Thank you for having me. So as you mentioned, I visited Allen Institute a few years ago, and that was the start of this project. And today I'm presenting how it evolved over time. The topic is the structural and cellular transcriptomic foundation of human brain disorders. And in order to go and look at the human brain disorders, we use the definitions by the Institute for Health Metrics and Evaluation, which has been doing this global burden of disease report since 1990. And every year there is a evaluation of the burden of different brain and non-brain disorders. And the way they define it is based on years of life lost and years lived with disability. They combine it and they define this DALY measure, which is the disability adjusted life years. You can see in the figure the DALY measure, and the way it works is the area for each disorder shown here is the percent that that particular disorder is contributing to the general DALY across the globe. The color coding for within rectangles is based on the type of disease. So red is for communicable disorders, green for injuries, and blue is for non-communicable disorders, which is basically the brain disorders that we will focus on. You can see the mental disorders in red box, neurological disorders in yellow, drug abuse disorders in orange, and a stroke in purple. And the color mapping is showing the disorders that are increased, has a positive rate of increase. So basically Alzheimer's disease, for example, has the highest between these brain disorders, highest rate of increase on the DALY measure across the globe. And if you look at the maps of these different type of brain-related disorders, we can see that they are widely spread across the globe, and there is a high degree of burden in North America and Canada as well, especially neurological disorders have been on the rise with the aging population. And this is until 2019, so the last 2-3 years might have changed some of these, but in general, there is an increase in the burden of brain disorders. So then we wanted to focus on a spectrum of different brain disorders. And what we need to consider is that the taxonomy of brain disorders have been changing over time. And at the beginning, this was based on the symptoms only, whereas the recent disease categories reflect both the root causes of the clinical phenomena as well as the symptoms. For example, neurodegenerative disorders or brain tumors are talking about what causes the disease, whereas motor disorders or psychiatric disorders are more based on the symptoms and the manifestation of the disorders. Using this approach and looking at brain disorders as a spectrum and looking across different diseases will help us to look at the shared unique and anything in between, that mechanisms that are involved in diseases. For example, the genetic overlap between different disorders can help us to understand the molecular pathways and biological pathways that are involved and how they can cause the pathophysiology of the disease. And these transdiagnostic studies are becoming more common because we have now more data, and a bottom-up data-driven approach is now basically possible. In particular, I'm showing one example study from Brainstorm Consortium in 2018, where they looked at the shared heritability and genetic correlation across psychiatric and neurological disorders. They looked at 22 different brain diseases from 265,000 patients and 784 control participants, and assessed the genetic correlation within each group, between them, and also between this participant across 17 different phenotypes. If we look at the genetic correlation across psychiatric disorders, we can see a lot of overlap. For example, bipolar disorder has a significant genetic correlation with major depressive disorders, OCD, and schizophrenia. Or major depressive disorder has a significant correlation with ADHD, anxiety disorders, bipolar disorders, major depressive disorders, schizophrenia, and Tourette syndrome. So there is a high degree of overlap between these different psychiatric disorders. But when we look at the neurological disorders, whenever we see overlap, it's mainly within the subcategories of neurological disorders. For example, epilepsy is correlated with focal and generalized epilepsy, or migraine is correlated with subtypes of migraine. So there is a more distinct genetic underlying pattern for neurological disorders. And when we look at the overlap between psychiatric and neurological disorders, we still see that neurological disorders are more unique and don't share as much genetic underlying factors with psychiatric disorders. Between different phenotypes and all neurological and psychiatric disorders, there is some degree of overlap. For example, we can see that years of education is significantly correlated with both psychiatric and neurological disorders, but it's positive correlation with ASD and bipolar disorder, and negative correlation, for example, with ischemic stroke. So with that in mind, we started our study, and the goal was to get these gene disease associations and investigate the transcriptomic patterns, both at the macro level and also at the cell type level. And to do so, we looked at the DisGeneNet dataset, which I will implement, and then we looked at different measures of gene expression, both at macro and micro level. And in each analysis, we looked at the differential expression and enrichment of these genes, the similarity of their pattern across different brain disorders, and then at the end, we compared the patterns between human and mouse models. One assumption that needs to be clarified here is that all the cases that we are looking at here are gene expression patterns that we consider normative. They're not from particular brain disorders data. And the idea is that if these gene expression patterns are happening in these normative tissues, the pattern will be similar, and only the changes in different brain disorders will happen within those cell types or brain regions that already express those genes. So this DisGeneNet dataset is a platform that has curated GWAS catalog and animal models and scientific literature data showing genes and disease associations. And we looked at all the disorders. We selected the brain disorders that are categorized based on the GBD report mentioned earlier. And we had 3 different selection criteria. We removed cerebrovascular disorders because the data that we use doesn't have gene expression patterns from vascular cells. We wanted each disease to be each gene disease association to be at least confirmed from 1 curated report. And each disorders that has less than 10 genes associated with it was removed. At the end, we ended up with 40 different brain disorders, 1,646 unique genes associated with these 40 disorders, and they were categorized based on the GBD into psychiatric, neurodegenerative, movement, substance abuse, developmental disorders, and brain tumors. And the 5 other disorders that didn't fit in these categories were put together in a brain-related group. The color code that I'm showing here will remain the same across the presentation, with psychiatric disorders as red, neurodegenerative disorders as yellow, movement as cream, substance abuse orange, and developmental disorders and brain tumors as blue and green. So the first thing we looked at was the uniqueness of the genes associated within each category, with diseases in each category. For example, there were a high degree of overlapping genes in neurodegenerative disorders, psychiatric disorders, and movement disorders. These happened within the category and also across all the other categories. For example, psychiatric disorders have a lot of genes that they share with substance abuse disorders and neurodegenerative disorders. Neurodegenerative disorders have genes that are shared with movement disorders, psychiatric disorders, and substance abuse disorders. But when we look at the individual diseases, this has a wide range of what is the percentage of the genes that are unique to that disease or is shared between that disorder and the rest of the brain diseases studied here. Next, we did a gene ontology analysis, and for that, we basically looked. Gene ontology analysis, for those who don't know, is a literature-based analysis that looks at the overrepresentations of a set of genes within a molecular pathway or a biological pathway, and is asking whether this gene set that we are looking at is more represented in this particular pathway or is at the level of random or underrepresented. And for that, we only looked at the unique genes from each disease category. We can see some of the pathways that we expect, for example, regulation of cell death and neuronal death in neurodegenerative disorders, central nervous system development and neurogenesis in developmental disorders. We also see regulation of neurotransmitters and modulation of chemical synaptic transmitters in substance abuse, movement disorders, and psychiatric disorders. There are also some more interesting associations. For example, we see defects in signal transduction in brain tumors, peroxisome dysfunction, which is related to demyelination and loss of white matter integrity, are related to developmental brain disorders. And we found the insulin IGF pathway to be overrepresented in neurodegenerative disorders, which has been recently shown that chronic type 2 diabetes is one of the underlying pathologies that can lead to Alzheimer's disease within the same pathway. Next, we looked at the gene expression at the macro scale. And to do that, we used the Allen Institute Human Brain Atlas. This atlas is based on the postmortem tissues from 6 donors. The samples were collected in 4 of the donors in one hemisphere, and 5 of the donors were male. In total, 17,348 genes were annotated from 3,702 tissue samples, which we pulled together into 104 structures. All the gene expression patterns were normalized across structures and across different brains. We look at the gene expression stability, expression patterns, and gene modules from this data set.
We have a question in the chat, yes. That's okay, from David. What is the rationale for categorizing autism as a psychiatric disorder category rather than developmental?
That was based on the GBD, but when we go in, and also when we see the categories, we can see that in the macro scale, it goes with other psychiatric disorders, but that is not necessarily the cut. And that is one of the main purposes of this study. So these categories basically put them in one or the other, but we wanted to see if we look at the spectrum where it fits, if that answers the question. Okay, I'll continue, and if there is a follow-up question, I will answer. So in order to look at the gene expression patterns, we needed to define the level that we want to look at. For example, we could look at each tissue sample separately, but there is a high degree of correlation between tissue samples. So first we looked at the consensus map that shows regional homogeneity of gene expressions. And to do so, it was based on the pair of regions, and the question asked is how many genes are differentially expressed between these pair of regions. And the definition is if they are basically there is a threefold change in expression level between the two regions, and it's happening at least in 5 out of 6 specimens shown before. The color bar here that goes from 0 to 12 is based on the log2 scale. So regions that are showing deep blue, basically they have really similar gene expression across all the genes, whereas the red color that shows, for example, is shown between white matter and cortical regions is 2 to the power of 12. So there are more than 4,000 genes that are differentially expressed between cortex and white matter. And based on this, we removed represented areas. So the 40 cortical regions were down, basically were sampled back to 8 cortical subregions. But this also can cause some overrepresentation. For example, we have 16 thalamic hypothalamus subregions because they have very distinct gene expression patterns. Then we looked at the differential stability of the genes, and the definition of differential stability of a gene is basically how similar is the pattern of gene expression across different brains. So we take 2 brains, we look at the expression pattern for a given gene, and we calculate the correlation, and then we average the correlation across 15 different pairs of brains with 2 out of 6 selection. And the correlation is basically anywhere between 0 and 1. The way it works here, you can look at the expression patterns for 4 different genes, and the red is higher gene expression and blue lower gene expression. For a highly stable gene like the DRD1 for dopamine receptors, we have a stability of 0.9, which you can see that the expression patterns across the 6 brains is very stable with high expression in basal ganglia, lower expression in cortex, but is still higher than average, and low expression in mesencephalon and cerebellum. When we go to MAPT, we have a moderate level of correlation. MAPT is the gene that is the underlying gene for tau protein, which is implicated in frontotemporal dementia and Alzheimer's disease. And we can see in one of the specimens, there is really low cortical expression. In another one, we have lower frontal, which 2 of the specimens have high frontal expression. And this can, if we look at the NGF nerve growth factor gene, then we can see basically there is no stability and the pattern can change from one brain completely to the other. And this is one of the things that we should keep in mind when we are looking at the gene expression pattern and correlating it with other phenotypes in the brain. When we look at the differential stability of the genes that are associated with different brain disorders, we found that in every single category, they have a significantly higher differential stability compared to the general genes that are not associated with brain disorders. And we also saw a gradient when we go from tumors to neurodegenerative disease to psychiatric and substance abuse. And the way it is we assume that it's because the genes that are associated with brain tumors and neurodegenerative disorders, they are more general functions. So they are expressed widely in the brain, but not necessarily with a pattern that is preserved across all brains. whereas genes associated with movement disorders, psychiatric disorders, and substance abuse, they are targeting a specific brain circuitries and a specific regional pathways. So they have higher stability. We can also see it at the individual disorders level with a lot of neurodegenerative and brain tumors disorders showing genes with really low stability, but each disease has also a spectrum within itself with genes with high and low expression stability.
We have
Next,
another question. That's
yes.
all right. From Malar. Go ahead, Malar.
Thanks, Yashar. Can you mind just going back a slide?
Yes.
This is fascinating stuff. I want to ask you a question about that middle plot where you just showed that gradient. So, for movement disorders, I would imagine a very stereotyped kind of pathway for degeneration and, as a result, gene expression, right? More so than kind of general psychiatric disorders, or even maybe substance use disorders would be more specific too. So, can you explain that kind of that ranking, or just think at a certain point, like at what point does this kind of idea of this gradient matter? Or would you kind of see this as more of like a bimodal distribution, I guess is what I'm asking.
I see it more as of a spectrum, but if you look at the plot on the right, that is actually showing what you are saying. So for example, for epilepsy, temporal lobe or ataxia, as they are up there, whereas only OCD from the psychiatric disorder is in the top 10 or in the top 10. But then you have spastic paraplegia. Sorry if I pronounce some of them because it's wrong. Or dystonia. Then you can see that there are some movement disorders that have I think they have a component of neurodegeneration that they have lower stability. So there is a more of a spectrum within movement disorder.
Right.
Whereas all psychiatric disorders are almost in the top 50% of the plot. Like the lowest one is schizophrenia, but you can see that almost 75% of the genes in schizophrenia still have higher than median stability.
That's interesting. So then if you kind of take that up a step further then, and you look at and you said schizophrenia is lower on this list, the way to get more specificity would be to somehow divide it up into component or into grouping. I mean, maybe this is exactly where you're going, and I'm ruining the conclusion.
This is we didn't do that for this particular study, but when we get to the cell type, that is the last component that I think we need to go in that direction, but we haven't done it yet, because then you can look at, even within neurodegenerative disorders or some of the movement disorders, you can still see variability.
Right.
So a disease like a schizophrenia, I think this one had like a few hundred associated with it. So you can imagine that there is a lot of variability, and I think grouping them into subgroups of genes will help a lot.
Great. All right, thank you.
No problem. So when we looked at the expression patterns and we tried to categorize the disorders into subcategories, which we call them anatomic disease groups because this is based on the broad anatomical expression pattern, we found 5 different categories, with brain tumors mainly in the ADG1, neurodegenerative disorders in ADG2, movement disorders, psychiatric, and substance abuse disorders mainly in ADG3. And then there are the rest of the disorders, they have a more variable pattern of expression, and they are categorized as ADG4 and 5. And if we look at it in more detail, we can see that one of the main factors that basically causes the categorization is the cortex. For example, even between brain tumors and neurodegenerative disorders, the main sub-brain region that has a consistent, significant difference between the two groups is the cortical regions, whereas there are some sub-regions within the subcortical structures that show difference. Group 3 has significant cortical expression difference with every other groups. We can see that group 3, 4, 5 have thalamic and cortical differences. But it's also interesting to see that some subregions of the brain, so for example, part of the mesencephalon and myencephalon, don't show any differential expression across these 5 different categories. The next question was, now that we have defined these, we have found these 5 clusters based on a hierarchical clustering, how stable they are. And to answer this question, we took the expression pattern of each disease from one brain, and then we looked at the other 5 brains and we asked what is the closest expression across these the 4D brain disorders to the expression pattern from the first disease. And when we found the disease to be from the same ADG group, we call it a success. You can see it as a solid color on the left. And then if we find the exact same disorder, then it's also a success for the unique recognition of the disorder. So ADG, across the disorders, we can see that there is a rather high level of finding the same ADG group. We have a higher variability when we try to do the same thing with the GBD group. For example, you can recover movement disorders rather stably and substance abuse disorders, but psychiatric disorders have a more spread confusion matrix where they get miscategorized with substance and movement disorders, while they are categorized in ADG3 within the same ADG group. So if we go further for this divergence and convergence within ADGs, we can see that there are disorders within these ADG clusters that have the stereotypical expression pattern. For example, glioblastoma and multiple sclerosis have a very similar pattern to the average ADG 1 gene expression pattern. Also, bipolar disorder and OCD have a comparatively stable and similar pattern to the average ADG3 pattern, with bipolar disorders having both cortical, basal ganglia, and thalamic expression pattern, whereas OCD has more cortical and the thalamic pattern. And if we zoom in even further, and here I'm showing ADG1 and ADG3 only, we can see that within ADG1, we have the frontotemporal dementia, Alzheimer's disease, and ALS with higher cortical expression compared to dementia and Parkinson's disease. And then we have basal ganglia expression in Parkinson's disease, but not in the other disorders. In ADG3, even the cortical expression pattern, which is very stereotypical for that group, can change. And this can partially explain why we cannot find unique genes when we try to look at different brains. So for example, you can imagine when bipolar disorders and depressive disorders or schizophrenia have a very similar pattern, why can recover the same disease category in the other 5 brains, they can get mixed up between the disorders. Yes.
So, I mean, again, very, very cool. One question I have when I look at this is, in the Allen Brain Atlas, you kind of have, relative to the amount of real estate the cortex takes up, the subcortical structures are somewhat over sampled. How much of have you been able to control for that in some way? And how much of that kind of subcortical and hippocampal, I'd say, how much of that oversampling might play into these results?
So I would go the other way. The question is how much overstudying we are doing in the cortical regions when it comes to the brain disorders.
Well, you don't have to convince me of that. I mean, that
I'm just the reason I say that is because if we have we still have a lot of variability when it comes, like, I'm going back to this. So we still have genes that are differentially expressed within the cortex and within subcortical areas. But when you look at the samples, there is really high correlations between them. And then the question becomes, maybe the factors that are defining why one region is affected in one disorders and not in the other might not be the underlying genetic factors. It might be the vasculature of the brain. It might be other factors that we need to consider. Whereas if a gene is really implicated in one disorders in subcortical areas, it might really target a subnucleus. For example, a good example is Parkinson's disease and substantia nigra, which is a small nucleus, but it's basically targeted both with dopaminergic genes and SNCA gene that is specifically highly expressed in that one region. So with that in mind, we actually try to make it balanced based on the gene expression, and we didn't try to make, because cortical regions have a higher area or surface, to make more samples representing the cortex. But that's the way we went. We can also change that. And a lot of the studies that have published with these data, as you know, they completely removed the subcortical areas because the expression was also measured with laser method rather than
a laser capture microdissection.
Yeah.
And so yeah,
because the
random sampling per subcortical nuclei, so it's like even more specific than that.
Yeah, so because of that, some people just remove those regions. I think the studies that showed like high correlation between brain networks and gene expression, they even removed hippocampus, and it was basically just cortex.
I mean, yeah, I mean, hippocampus is kind of important in a lot of these disorders, maybe.
Yeah. No, I agree.
Yeah, thanks for that description. Very helpful. And I agree with you. I think it's and this is a nice way to integrate those structures into this analysis, where, like you say, often they're completely ignored. So well done.
Yeah, and then now this slide, that is actually a good transition to go to this slide, because then we looked at the canonical gene modules that was based on the WGCNA analysis. And the way it works is basically it's trying to find the co-expression of, based on the gene co-expression, it tries to make them in subnetworks of genes that are expressed together across all these samples. And to do so, basically, the study used only highly stable genes, the top half, and then they identified 32 different modules. And now we can see we are going from a regional space to a gene space. So instead of caring how many samples we have in cortex or subcortical areas, now we are asking how much convergence and how much co-expression we have between different genes. And if we look at these 32 modules, which are sorted based on their neuronal content, we can see that a lot of them are expressed widely across cortex and subcortical areas, but a lot of them are also completely absent in the cortex. Some of these modules are targeting very specific regions in hypothalamus, in midbrain or striatum, whereas some other modules might be expressed across multiple regions, which is in line with the homogeneity of those particular gene network across those regions. And if we try to look at the brain disorder expression based on now these modules of genes, we can see that the ADG1 group, which was mainly the brain tumors, is now expressed in M21 to M32, which has a high glia content, whereas ADG3 is mainly expressed in M1 to M10, which has a lot more neuronal content. Although these modules can be cortical or subcortical. And then ADG2 now has a lot more variability, being both implicated in neuronal and non-neuronal modules. ADG4 and 5, which have a lot more variability between them, target specific regions. For example, marijuana abuse is expressed in M10 within a striatum. M11 with thalamus is implicated in Parkinsonian and tobacco use, and M12 with substantia nigra is expressed in Parkinsonian and alcoholic intoxication. The other thing that these gene space versus brain structure space helps us with is identifying unique disorders across the 6 brain. So if we repeat our analysis for the stability and success rates of identifying gene expression patterns based on a given expression from one brain, we can see that we can recover now across all the ADG group much better, uniquely the same disorder instead of a similar disorder. So it seems that we are now identifying the pathways that are implicated within each disorder better, and they are more unique. And this provides a bridge to go to the cell type analysis that we did, because these gene networks are more specific to cell types as well. And as well, the way it works is when the samples that so far we looked at, each sample is basically an average expression across all the cells that are included in that sample. And that makes comes from some issues. For example, if you have cell types that are underrepresented in each of these samples, but they are implicated in a disease, their signal will be averaged out and we cannot distinguish the changes in those. And later on, if we look at other samples, if we see upregulation or downregulation of expression in certain regions, we don't know what brain circuitry or what underlying cells are causing that all upregulation or downregulation. And while we have some level of a special specificity, it's not at the cellular level, it's more broad that all the genes within these samples have that. WGCNA analysis helps with that, because now we are looking at the genes that are changing together, and supposedly we can assume that they are changing together because they are coming from particular cell types. And the eigen genes are more functionally specific. But when we move to single-cell RNA-seq data, we can look at the cellular resolution. We can now study the underrepresented cells, because if even we have 100 cells out of thousands of cells, we can look at the expression pattern within those cells. And we can look at the expression and co-expression patterns and look at their functional importance. So with that in mind, we moved and used the Allen Institute MTG dataset from human middle temporal gyrus. There were 8 donors, 4 female and 4 male, with the age range from 24 to 66. The samples were surgically removed, and in 3 cases, they were from epileptic patients. However, it was assessed, and the assumption is that the cell types represented here are representing the normative cell type. And in total, 16,000 cells were isolated. And by cells here, it's a bit different. These are nucleus, because cell isolation in human is a bit more difficult than animal models. So they went with single nucleus RNA sequencing. And when they did their clustering based on the gene expression patterns in these 16,000, they found 45 inhibitory, 24 excitatory, and 6 non-neuronal subcategories. I'm showing the TSNE maps that they provided in their study. And the way the TSNE works is basically you're bringing this high dimensional data from the gene space and the co-expressions into a 2-dimensional space. And you can see there is a high degree of separation between excitatory, inhibitory, and non-neuronal cell. There is a high overlap between donors. So basically we are not catching a lot of donor specific cell typing, and the cell types are coming from the biology. And you can see within each cell type a gradient based on the layers that they are expressed in. So for the rest of the study with using this data set, we focused only on 24 diseases that they had higher than median expression in cortex based on the macro scale data. The first measure that we looked was the equivalent of the differential stability, which is cell specificity. And the measure we used was tau, which is trying to answer the question is how specific is a given gene's expression for a given cell type. And if the gene is uniformly expressed, basically the tau will be 0. And if the gene is only expressed in one cell type, it will be 1. Anything in between can be between 0 and 1. If we want to give another example, for example, for a gene that is only expressed in 3 groups is around 0.95 tau expression. Again, we see a significantly higher expression level for the genes that are associated with brain disorders compared to the general genes that are not associated with brain disorders. We see a similar gradient. And now, if we go back to your question, you can see that movement disorders here are much more specific because cell type specificity is a better measure to capture the targeted cells. And that is like, I would say, followed by psychiatric but substance disorder, but with a higher drop. And they are more similar to neurodegenerative and brain tumors here. Again, we have a high degree of variability across within each disease, as you can see on the plot on the right. So we repeated the clustering for these 24 disorders, and we found 4 different cell type groups, with one group being astrocytoma, which is highly expressed in non-neuronal cells, and the rest of the disorders being categorized in CTG1, CTG2, and CTG4. CTG4 is mainly the neurodegenerative disorders that had higher cortical expression, and they have both non-neuronal and neuronal expression. And if we further look at the differential expression between these groups in group 1 and 2, we can see that the differential expression comes from both inhibitory and excitatory cells, as well as one subgroup as well as astrocytes. And we see a significant difference between group 4 and both group 1 and 2 in non-neuronal cells, specifically microglia, as well as the astrocyte cells. There is also a gradient in inhibitory cells going from CTG1 to 2. There is an increase of expression within inhibitory cell types and a decrease in excitatory cell types. So next, we looked at the mesoscale cell type consensus map, and the way we did this, we used UMAP. It's a similar embedding of patterns in a lower dimension. And we averaged the correlation matrices for both structural and cell type data, and we applied the UMAP on that. And as you can see, now we can see in these 24 disorders, we go from group 2, which was mainly neurodegenerative disorders, to group 3 in ADG, and we go within that spectrum, we go from non-neuronal to and glia-specific cell types to the disorders that have both glia and GABAergic cells, and then going to more glutamatergic expression within disorders. We also looked at the cell type specific expression and compared it between human and mouse. To do so, we included the cell type data from Allen Institute for mouse primary visual cortex V1 and motor area ALM from 23,000 cells. And the way the homology between these cell types were identified was that the RNA-seq data was put together. There was an unsupervised alignment between these expression patterns, and then the clustering was done for all these 23,000 plus 16,000 data together. As you can see, the level of co-clustering when you separate the two groups and look at the matrices, you can see, for example, the PAX6 expression in human is expressed in the PAX6 from mouse is expressed in 2 different cell types from human. You can see the p-valve cell type in the inhibitory neurons is basically co-expressed across these cell types in human and mouse. And you can also see the layer specificity preserved between the 2 species. Based on these alignment, there were 32 different homologous neuronal and 5 non-neuronal homologous cell types identified. We further averaged the VIP and SST groups subcell types to one big category, and we came up with different subcell types that are homologous. What we did next was we take the average expression patterns for each disorder in human and in mouse, and we cluster them together. And you can see that there is a high degree of similarity in the pattern. And in fact, there are a lot of hit points that the two disorders are the closest to each other when we go for clustering, including, for example, ataxia, epilepsy, Alzheimer's disease, bipolar disorder, schizophrenia. And some diseases have a degree of separation that is not as far from the other one. For example, autistic disorder only is more similar to substance withdrawal syndrome within mouse than the autistic disorder in human. But then depression and alcoholic intoxication show further apart patterns in human and mouse. We also looked at the enrichment, which is different from the expression pattern. Here we are looking at the level of expression, and we are asking if for a given gene set, the expression is higher than random. And we use the EWCE method, which is basically comparing the expression level of a given gene set to 100,000 permuted gene sets with similar lengths. And the results are FDR corrected. And here we see much more divergence between human and mouse cells. So while the expressions patterns might be similar, the level of expression is not the same between the two species, and the hit points are different when we look at inhibitory and excitatory cells. And just for the last part of the presentation, I'm looking at 3 different disorders that have been mainly categorized together. So autism, bipolar, and schizophrenia were very close, showed very close expression patterns at macro scale, at cell type level, and also when we look at the mouse and human data, they are clustered very close to each other. The question that we were trying to ask was the pattern of co-expression within different cell types for genes associated for these disorders, and which we wanted to see which cell types co-vary together within these disorders and what genes are underlying this covariation. But in order to remove any biases, we removed all the overlapping genes. So we are only looking at the genes that are unique to each of these 3 disorders. We looked at the covariation across cell types based on a cosine transform and thresholded it at 0.5. So if you have a higher than average co-expression, and you can see that while there are co-expressed cell types within autism, bipolar, and schizophrenia and inhibitory cells, there is a lot higher expression and overlapping co-expression within excitatory cells. And this happens across different layers for excitatory cells. Then we selected the genes that were both in inhibitory and excitatory cells with higher than 1.5 standard deviation expressed within each cluster of co-expression. And now we are looking, we did a functional enrichment analysis, Go Ontology analysis, and we were looking that while we started with completely unique genes, what pathways are enriched. And we can see that we find a high degree of overlap in the functional enrichment between bipolar and schizophrenia, but not as much with autism. And most of these pathways come from regulation of synaptic signaling and the chemical synaptic signalings between the two. However, there are some unique aspects. For example, the genes, some genes in bipolar disorder, a lot of genes actually are associated with circadian behavior, sleep-wake cycles, and basically cyclical functions of pathways that are implicated in cyclical functions. For autism, we see that unique genes here are involved in brain development, health development, and social behavior, which is what we expected as well. To summarize what I showed you today is we looked at the macro scale gene expression pattern, average gene expression patterns for 40 different brain disorders, and we found 5 primary anatomic group. And while these anatomic groups tend to associate with groups from GBD together, we have exceptions, and we think that if we now look at those exceptions, we might find underlying pathways that are actually informative in terms of understanding what are the shared mechanisms between these disorders that are grouped together. A large majority of brain diseases exhibited a brain-wide transcriptomic pattern. However, these patterns could be retrieved within the ADG groups. And when we added the WGCNA modules and cell type modules, we could even find the specific disease pattern across the 6 specimens and also different donors in the cell type. Genes associated with the brain diseases have both higher differential stability and cell type specificity, and we see a gradient of the specificity and stability when we go from neurodegenerative disorders toward psychiatric and movement disorders. The largest component of transcriptional variation was in non-neuronal and cortical regions. And non-neuronal expression variation is also identified in the cell type data, even though we focused on the cortical disorders. We also saw a higher variability in inhibitory and interneuronal cells. And finally, disease expression patterns were largely concerned between mouse and human. However, the enrichment level was more divergent between the 2 species. Finally, I wanted to thank everyone who came today to this talk, as well as Mike Havrilisha specifically, which helped a lot with this study and guided me through the study, as well as my other collaborators from Allen Institute and also Iowa University. Thanks.
All right, thank you so much. That was a really, really great talk. It was super interesting. We've got time for a few questions. So if anyone has some questions, just go ahead and write them in the chat or raise your hand. We have a first question from Etienne. Do the removed overlapping gene expressions also show mostly excitatory activity?
The patterns are similar, yes. So when I don't have the figure here, but we have it in the supplementary figures of, and they also show that the patterns remain largely similar though when we remove the overlapping genes. The only thing is when you want to look at the overlapping genes, removing overlapping genes, you need to look at every 2 diseases together, because if you remove overlapping genes across all disorders, then again, that is not as representative for each disease.
I have a quick follow-up question, if that's okay.
Yes.
When you classify...
1 second. Is it okay if I stop sharing so I can see people who are asking questions?
Yeah, of course, go for it.
When you classified in inhibitory and excitatory, do you refer to the gain or loss of function that the gene expression gives, or is it excitatory versus inhibitory neurons?
No, it's only the cell types, and here again, the limit. This is one of the first assumptions that I was saying in slide 4. These we don't see any downregulation or upregulation related to the disease. We only see the expression level related to the cell type based on the clustering of the cell types. And the cell type clustering has been validated with histology and also markers that are more well known as well. So it's mainly the categories, excitatory and inhibitory cell type categories.
Okay, thank you. Great study. Thank you very much.
Thank you.
There's a question from Johan.
Hi, yeah, this is all very cool stuff. I'm wondering, can you comment on whether the disease overlaps that you see here are reflected in the comorbidities that you would see in the clinic? So for example, with diseases that are kind of closer together in that consensus representation you showed, are individuals more likely to be diagnosed with one of those diseases? Would they more likely have a comorbidity that's sort of closer in that latent space?
They are, but this is not based on this study, and I'm not looking. We didn't look at that, but there are studies that they are looking at genetic correlation and the comorbidity and the shared heritability, and they show that there is a higher degree of shared phenotypic manifestation for the disease that they have higher shared heritability and higher genetic correlation. But interestingly, that also happens more in psychiatric disorders, and it's at least studied a lot more in the psychiatric disorders. Whereas when you look at the neurological disorders, it's not as much the case.
Thank you.
Are there any other questions? Okay, if not, I think we can head straight to the student discussion. So thank you very much again, Yashar, for this great talk today. This concludes today's lecture. So as I mentioned earlier, anyone who wants to can stay for the student discussion, and a great rest of the day to everyone else.